Hello everyone, welcome to AG Biology. My name is Arvind Gopi. Today in this video, we're going to discuss about antibiotics, its actions and mechanisms. Is one of the key areas in which CSIR asks too many questions. So let's get into the video. First of all, where does antibiotics or antibacterial acts on? It acts on key essential pathways and processes that happens in a bacterial cell. First of all, cell wall synthesis. Cell wall synthesis specific for bacteria are targeted by these antibacterials, for example beta-lactam antibiotics. In addition to cell wall, cell membrane is also targeted. For example, polymyxin B, which acts like a detergent action. Another pathway which is targeted is folic acid synthesis. Folic acid trihydrofolate is synthesized through an intermediate dihydrofolate. In each step of these reactions, there are antibacterials, for example, sulfonamides and trimethoprim. Now let's look at the key information pathways. In information pathways, DNA is replicated by itself into another molecule by means of DNA replication, and then it is transcribed to form an intermediate molecule, messenger RNA, by the process of transcription, and finally this gets translated by the process of translation. In each steps, antibacterials can be used to target a particular process. For example, in case of replication, topoisomerases are very important. So quinolone antibiotics acts on DNA replication. Now if you look at translation, you see a plenty of molecules being synthesized or isolated, which either targets larger subunit of the ribosome or smaller subunit of the ribosome. So let's see some of them in detail. Firstly, I told you in information pathways, DNA replication, there is yet another enzyme which is key for bacterial cell to replicate to maintain its supercoiling state. That is nothing but topoisomerases. So topoisomerase inhibitors can be used as antibiotic. For example, nalidixic acid and ciprofloxacin. What is the key topoisomerase which acts on replication? Yes, that is DNA gyrase. DNA gyrase has two domains. One is gyre, which does the breaking and resealing, and then another domain which is B, gyre B, which does the energy transaction. Novobiazine is the molecule which targets the energy transaction, whereas ciprofloxacin, nalidixic acid, like the quinolone and fluoroquinolone derivatives, target the gyre A domain. Next is rifampin. DNA-dependent RNA polymerase is a key enzyme involved in transcription. So it acts directly by blocking the RNA polymerase transcription. Now if you come to translation, you see plenty of molecules. One is tetracycline. Tetracycline binds to the smaller subunit and then interfere with binding of the tRNA. Yet another molecule which binds to the smaller subunit is aminoglycosides. They bind to smaller subunit and cause mRNA codon to misread. There are plenty of molecules like chloramphenicol, linezolid, macrolide antibiotics. These binds to the larger subunit of ribosomes. So one special molecule which acts on a very different style, for example puromycin, which terminates the translation by mimicking a tRNA. I mean a charged tRNA in the A site. So it makes a peptidyl transferase reaction and forms peptidyl puromycin, and thereby forming a peptidyl puromycin, it blocks the reaction. Chiromycin and fusidic acid. This blocks the elongation factor Tu or elongation factor G. Remember, these elongation factors that we see in translation are able to bind to GTP. So these are preventing the GTP association, for example, chiromycin, or in the case of EFTU, fusidic acid in the case of EFG-GTP. Now let's try to answer one question from CSIR previous year. An antibiotic that resembles the 3' end of a charged tRNA molecule is? Yes, we know that it is puromycin. It mimics a charged tRNA at the A site. So answer is C, puromycin.